Hi guys, this is Jess over at Absinthe Reptiles, and today I was going to show you some basic leopard gecko genetics. Hopefully this will help some of the newer people trying to get into breeding and they don't understand what will happen if they breed a normal to an albino, for instance. So I'm hoping to help people out like that. Alright, so let's get started. Oh, and also, most of these pictures are mine. I do have one picture that came from Peach Geckos, and she so lovingly let me use her beautiful super smell. But we'll see that sooner or later, but there's only a couple pictures that are not mine, and I am going to post the links on the description, so I am not claiming these as mine. But okay, so first we're going to start with albinos, and it is a recessive trait, meaning that you need the allele on both parts of the paired chromosome for it to express. So, but if you have a different allele next to it, the dominant allele is going to show because it's recessive and it doesn't have the pair. So the dominant allele will always show through if it's not paired. And just to have some fun, maybe do some examples if my computer will work for me. So we have two Trimper albinos. This is my girl actually. So we're just going to say small t is the Trimper gene, and since it's paired, it needs another small t. So both of these will be small t's. So for a Punnett square, whoops, and a big T. I lost my pen. Here we go. They both need to have small t's on the Punnett square. Hopefully some people know how to do this. I know I learned how to do Punnett squares, I think in 8th grade. But because they're all small t's, you're going to get 100% Trimper from a Trimper pair. For the next one, I have a normal and a Trimper. So once again, the Trimper is going to be two small t's, and the normal, since we're going to say it does not have any albino, we're going to probably get to that in a second. Yeah, we are. We're just going to say that it is completely normal, no het. So it'll be big T for the dominant normal allele. So we're going to put the Trimper here and the normal over here. So it's going to have a big T and a little t in all of the offspring, which will give you 100% normals, but they will be het for Trimper. And het just means heterozygous, which means it has the normal and Trimper albino gene in it. So let's just say I breed two hets together. Like these two came from that pairing before, so it's going to be big T, little t for both of them. So then you're going to put a big T and a little t here. So over here, it would have two big T's. Right here, it'll have a big T and a little t. This one would be big T, little t, and little t, little t. So you're going to have 75%, and I'm just going on phenotype, and we're not going to talk about genotype. That's just going to get a little crazy, other than saying het. But we're going to have 75% normals from this pairing, with a 25% chance of a Trimper albino, or whatever albino that you're doing. We're just using Trimper since my girl's Trimper. But anyway, speaking of types, there is 3 types of albinos. Move this over. So we've got the Trimper albino, which is the most common, at least I've seen. Next one would be Bell. And then there's also Rainwater, which is also called Las Vegas albino. So that's that. But what the thing is with these types, though, they are not on the same part of the allele. So let's just say we took my two here, and of course, she is a Trimper, and he is a Rainwater. Because these are on different parts of the allele, whenever you breed these two together, even though you have two recessives, it's not going to show as an albino. It's going to come out to be 100% normal. Oops. So the thing is, with these guys, it's good to try to keep the albino strains separate. There's been rumor that having both albino strains tends to cause some genetic issues. At least I've heard. There's also rumors that it's just causing confusion because it's kind of hard to tell the difference between them. So it's just recommended that you do not mix the albino strains because of this. Okay, and the next one we're going to talk about are polygenic traits. And a polygenic trait is basically where you take a trait that you're trying to breed for and you breed it, like let's just say we're going to try to make an orange animal. You're going to take your orangest animal and you're going to breed them together to try to get another orange animal. So it's basically line bred traits. So first we're going to talk about hypo. And with hypos, it just means hypomelanistic. And melanin is this dark pigment that you see, like the spots. And that's just to help absorb light. So in order to be considered hypo, they have to have less than 10 spots on their body. This excludes their head and their tail. So if your gecko has 5 spots on its back, like this girl, I think she has 5, then it's considered hypo, but any more than that, it would be considered high yellow. Next is super hypo, and these guys have absolutely no spots on the body, excluding the head and tail. So this girl is a hypo, so she has nothing on her, no freckling, no nothing. Tangerine is just a gene that people breed for oranging, like just the orange body. So I don't know what would make you really consider a tangerine. I guess it's just if it's orange. Next one is bald or baldy. In order to be that, you must have absolutely no spots on the head. There cannot be any freckling. There can't just be one spot. It has to be completely gone, which hence the reason why it's called bald. Next is carrot tail. And carrot tail is just the orange on the tail. And in order to be considered a carrot tail, it must have 15% carotene. And these girls, the top one has 25 or 27%, the bottom has 33%. I've calculated them before, and I'll show you how to do it. So first you're going to take the length of the tail, like the entire length. I try to start from the end of the point of the hips. Usually you can tell where that is. And then you're going to take the length of the orange. So with Nirvana, this is her tail, my tangerine hypo or super hypo. She had only 1 inch of carotene, and her tail is 3 inches long. So you put the length of carotene over the length of the tail, and I got 0.33. You're going to get a decimal because the carotene is going to be shorter than the tail. And then you multiply it by 100% and it'll give you the percentage. So she had 33% carotene. Next is patternless traits. I'm only going to talk about, I think, 2 in here. I'm not even going to try to get into stripe patternless. I barely understand the jungle gene itself. So anyway, sorry, my computer's messing up. Okay, it's a recessive trait, just like albinism. So it needs to have the paired chromosomes, and it has to be the 2 recessive traits. The first one is Murphy's patternless. This girl used to be mine. She took off. Never could find her. It's been months. Don't know where she is. But it, whenever they hatch out, they tend to have these raindrop spots, and they'll disappear as they grow. So usually you'll find this out if they're Murphy's patternless when they hatch. Next one is the blizzard, which is, I think, the most common of the patternless traits I've seen. And this gene just breaks out the pattern and it disperses the melanin throughout the body. And it'll kind of give the gecko a smoky color. But with this guy, he's a super snow, so he kind of looks lavender. I've seen some lavender normal blizzards. And another thing with blizzards, they often experience a genetic glitch in the eyes known as blizzard eyes, so they'll show up to have eclipsed eyes or marbling or snake eyes, but this really isn't that gene, because if you try to breed for that, it's not going to show up. It's just a glitch that happened with the blizzard gene, so that's not a thing. Next is blazing blizzard, which is just the albino version of the blizzard, and the albinism takes out the melanin and it leaves a white or yellow animal, which is why he's kind of whitish and yellow. Next one is Max Snow, which is really common. And this I believe it's incomplete dominant, but I've heard it's codominant. But the way I've learned it and seen it, I'm going to teach it as incomplete dominance, and I'm going to show you why. And usually whenever you go into school and you're first learning about genetics, you get this, this punnett, whenever he grew peas, he bred a white flower to a red flower, and he ended up getting a pink flower. And this is because the genes were incomplete dominant. They couldn't cover one of each other. So it ended up mixing. And that's basically what this is. And the homozygous form, which when I say that, that means it has both pairs and they're both the same, so it doesn't have a dominant allele and a recessive, it's just a dominant. So that's what these white and red guys are. They're homozygous, and this one would be heterozygous. And I'm only going to explain that because of the next thing. The homozygous form of the max snow trait is the super snow. So these guys will have the white body and the black spotting, and they'll usually not have any yellow at all. So that's what a super snow looks like. The heterozygous form, is just the plain old max snow. And these guys will kind of have the yellowish body with some lavender in there, and etc. So whenever you take a super snow, which we'll say is the white flower, and you breed it with a normal, which will be the red flower, because it's technically the homozygous, or the opposite homozygous of the super snow, and you're going to breed those together, and the pink flower is just going to be the normal max snow, and you can see where they kind of mix together and make that animal. And we're going to do a couple punnett squares, and max snow is kind of difficult. So we're going to say since this is the heterozygous form, it's going to have a dominant and a recessive. So we're going to say the max snow gene, which is the super snow, will be a big S, and the normal will be a small s. So for both of these, it'll be that. So it'd be big S, small s, etc. And then we're going to have two big S's. So with this pairing, breeding a max snow to a max snow, you're going to get a 25% chance of getting a super snow. And then it's going to be a 50% chance of just another max snow. Oops. Sorry, my tablet doesn't want to work for me. And then you're going to have a 25% chance of just a plain old normal. But with these normals, even though they came from a max snow line, if you bred them to another normal that came from the same clutch, for example, you're just going to get full-on normals. They do not have any max snow in them at all. So the next pairing we're going to show is max snow and a super snow. And this is kind of funny because both of these beautiful ladies are ladies, but just for example purposes, it's going to be the same thing. This is the heterozygous form, and this is the homozygous form, so it's going to have two big S's. So we're going to put that in. Big S. So with this pairing, you're going to get a 50% chance of super snows and a 50% chance of just a regular max snow, so there's no normals coming out of this pairing. Then when we go to the both homozygous forms, which I'm sure you guys have already figured out what will happen here. So this one has two small s's, and this one's the big S. And this one just means that it has absolutely no max snow in it. So try to make it distinct here. With this pairing, you're going to get 100% max snows. Alright, well that's all I have. Those are the most common ones I've seen so far. But if you guys have any that you want me to show, I can try to do a video on them. I know some people wanted to see clips and all that stuff. But if you have any suggestions, just leave them in the comments below or message my Instagram account that I have up. Anyway, this was just a little show to maybe help out people, and hopefully it did. Once again, this is Jessica from Absinthe Reptiles, and I'll see you guys in the next video. Bye!